Friends, welcome to Biology Made Easy. Today in this video, we'll discuss multiple allelism. We know in Mendelian genetics that each factor or gene has two contrasting alleles, and any one of the alleles can be represented by a gamete. But when we study a big population, it is seen that sometimes in the population more than two alleles are present. Again, you remember, in a person there may be two alleles, one dominant, one recessive. But when we study a big population, there may be more than two alleles. In a population, that is important. More than two alleles are present, that is known as multiple allelism. So how this multiple allelism usually arises? Usually it arises due to repeated mutation in different directions. That is the source of origin of this multiple allelism. So let us discuss about the different aspects of this multiple allelism. In multiple allelism, we have seen that in case of human being, there are three alleles. Three alleles in case of human A, B, O blood group. There are four alleles in case of the coat color of rabbit, and there are 15 alleles in case of eye color of Drosophila. That means in all these populations there are more than two alleles. Now, when more than two alleles are present, what are the different characters of this multiple allelism? Now, the first thing, number one, that is they are mutants. They are mutants of the same gene. That means the same gene has been mutated in a different direction. That is one of the important characters. The second important character is they are present on the same locus of same chromosome or homologous chromosome. Then another important point is in a gamete only one allele is present. In a gamete only one allele is present, and the different alleles, they represent different alternatives of the same character. Multiple alleles represent different alternatives of the same trait. That means the trait or character is the same, but different alternatives are there. Then another point is that sometimes they represent co-dominance. When multiple alleles is there, sometimes they represent co-dominance, as in the case of AB blood group in human beings. Or sometimes they represent dominant recessive nature, as in case of AB blood group in case of human beings. So these are some of the characters of this the multiple allelism. Now let us take the example of this multiple allelism, and the best example is the human blood group, that is ABO blood group, which we will discuss in detail. So the ABO blood group has been studied by Lance Taylor. So let us take the example of ABO blood group. in humans. So this has been studied by the AB blood group has been studied by Lance Steiner, but the AB blood group that is studied by Castellano and Sturley. Now this AB blood group is not only present in human beings, they are also present in case of primates, but not in monkeys. So from this one thing is clear that the primates are our ancestors, not monkeys, because usually we have the concept that monkeys are our ancestors, but from the blood group it's seen that the primates are our ancestor, not monkeys. This is one thing. Now besides this ABO blood group, there are also other blood groups like the MN blood group, then Bombay blood group, then RH blood group, then P blood group. All these types of blood groups are there, but these are not related with multiple allelism. That is very important. These are not related with multiple allelism. Only the human blood group is related with multiple allelism. Now let us discuss the genetics of this ABO blood group, how it functions. The ABO blood group is controlled by a single gene. Let us study its genetics. So it is the ABO blood group is controlled by a single gene I, and the I, it stands for isohemagglutinogen. Isohemagglutinogen. I repeat it for this. Now this gene I, it has three alleles. And what are the three alleles? The three alleles are IA, IB and I. These are the three different alleles. But in any organism, only two of them will be present, not all the three. In different organisms, different will be present, but all the three will not present in the same organism, only two will be present. That is again important. Now, this IA, when this gene is present, it produces antigen A, which is a glycoprotein and is alpha D galactosamine, and it gives rise to blood group A. When IB is present, it produces antigen B, and this is a glycoprotein, that is alpha T galactose, and it gives rise to blood group B. And when small I is present, it produces no antigen. Again, this is important. It produces no antigen, and it gives rise to blood group O. Now, out of these three alleles, now this IA and IB, these two are dominant over small I. These are dominant. And IA and IB, these two are codominant. So now we can express it like this: IA is equal to IB, but both of them are dominant over I. Now when these different alleles will be present, how many genotypes will get? So usually the common formula for genotypes is, it is X x into x plus 1 divided by 2, where x is the number of alleles, x is equal to number of alleles. So here it is 3, that means 3 plus 1 divided by 2 means it will be 3 into 4, 2, that means it will come into 6. That means out of these 3 different alleles we can get 6 different types of genotypes, and these 6 different types will give rise to different types of blood groups. Now let us study in detail. So if we'll study it in a tabular form so that it will very much clear to you and very easy also to remember. Now suppose the allele from parent 1, then allele from parent 2, then the next will be what will be the genotype, then antigen, then antibody, and then the blood group. Now suppose in the parent, first parent, it is the allele is IA, and in the second also it is IA. We are discussing different probabilities and how many genotypes are coming. So in this case, the genotype will be IA and IA, means it is homozygous, and here the antigen will be A, and the antibody in plasma, will be B, and the blood group will be A, because antigen is A. Now if it is IA, but this is IB in the second parent, then the genotype will be IA, IB, and here the antigen will be both A and B, and in the plasma there will be no antibody, and here the blood group will be AB. Now take the third example. In one parent is IA, in the second one it is a recessive one. So in this case it will be IA and small I. So in this case it will give rise to antigen A and antibody is B, and here the blood group is A. So we have taken all the probabilities with IA. Now let us take the probability with IB. Now if it is the first parent is IB and the second one is IA, so it will be IB and IA. So in this case the antigen will be A and B, and there will be no antibody, and the blood group will be AB. Now the first is IB, the second parent also IB, so it will be IB, IB, and in this case the antigen will be B, here antibody B, and here this is A, and here the blood group is B. Now second, it is IB, now this is recessive I, so in this case it will be IB and I. So in this case also the antigen will be B, and this will be A, and it will be B. Now the next one, suppose it is small I, then it will be small I. So in this case there will be no antibody, but the plasma it will have both A and B, and the blood group is O. So you see how many types of genotypes are there. The genotypes are, they say IB are twice are there. Otherwise 1, 2, 3, this already we have taken 4, 5, 6. That means there are 6 genotypes. And blood group how many types? A, B, AB and O. So if you will remember this table, easily you can solve most of the questions. Any question is asked, easily you can solve. Now let us take one example from this, how these blood groups are inherited from parents to offspring. By looking into this, easily we can solve it. Now let us take one example, the inheritance pattern. inheritance. Now let us take one example, that is first example, homozygous A and the other parent is also homozygous B. That means one parent is homozygous A, the other is also homozygous B. That means it will be IA, and this will be IB and IB. So in this case, what will be blood group the offsprings? Easily we can write AB. Now, either you can write in this way or you can write it with the genotype. Now it will be AA, it will be AB, it will be AB, and this will be BB. That means in the offsprings, how many types of blood groups will be found? Oh, sorry. This is, we have taken homozygous, so it will be AA and BB, because it is homozygous. So it will be AB, this is AB, this is also AB. That means in the offspring, we will get only one type of blood group, that is AB. That means the offspring will have only AB blood group, because both are homozygous. Now let us take both the parents heterozygous. Second example. That means heterozygous A. So heterozygous A means it will be IA and small I. And heterozygous B, that means IB and small I. It is homozygous, that is heterozygous. So in this case, what we will get? Again you see, you can write in any way. We can write AB or we can write in this form, any way you can write. So we write IA and small I. This side will write IB and small I. So in this case, this will be IA, IB. This will be IB and small I. This will be IA and small I. And this will be small I. So you see, when this one, it will give rise to blood group A. This will give rise to blood group B. This will give rise to blood group AB. And this will give rise to blood group O. That means the offsprings have any one of these blood groups. It may be A, it may be B, it may be AB, or it may be O. Any type is there. Now let us take also third example. Suppose the third example says both parents are AB. Both parents are AB. So very easily you can do like this, A and B. So AA, AB, then AB, and this is again, this is BB, this is BB. That means what type of bloods we are getting here? We are getting blood group A, this is A. This is B, we are getting B, and these two cases we are getting AB. That means when both the parents are AB, the offspring can have A, B or AB. This very easily you can do. We will take a fourth example. The fourth example, let us take one parent is AB, the other parent has O blood group. O. So in this case we can write AB. And this side we can write OO. So this will be AO, this will be BO, this will be again AO, this will be BO. That means in this case there will be two blood groups, only A and B. That means here only we will get A, B blood groups. So if any question is asked, what is the inheritance pattern of these genes? Either you can do like this or you can take the genotypes by looking into this table. So this is very important and important for examination point of view. Now another important thing is, Let us discuss the blood transfusion. When one requires blood, what type of blood can be transfused to it? That is again very important. So let us discuss it here. Blood transfusion. Because this is important, because any person, any part of the life may require blood. So what type of blood groups can donate or can accept, that is important. So in blood transfusion, usually there are 4 types of blood groups: A, B, AB and O. So let us write also in this side A, B, AB and O. And this side let us write donor, and this side let us write acceptor. Now A can donate blood to A, no problem. B can donate blood to B, AB can donate blood to AB, O can donate blood to O. There is no problem. Now what are the other probabilities? Other probabilities, the O can give rise to blood to AB, it can donate blood to B, it can also donate blood to A. Similarly, the AB, it can receive blood from B, it can also receive blood from A. That means the O blood group is universal donor. Why universal donor? Because there is no antigen. Because in the blood there is no antigen, so it is universal donor. While this AB group is universal acceptor. Why universal acceptor? Because it has antigen A and B. Both A and B antigens are there, so it can receive blood from any group. So AB blood group can receive blood from anyone, while O can donate to anyone. So this is known as blood transfusion. Then besides this multiple allelism in case of human beings, there are also some other examples, and we can take one example from the coat color of rabbits, and also we'll take one example from plants, and that briefly also we'll discuss. So, in case of a coat color of rabbits, there are 4 alleles, and all the 4 alleles then do not function the same way. There is a degree of dominance. Some alleles they are more dominant over others. So, if we take that example, that is example number 2, that is coat color in rabbit. So in this case, usually what happens, there is if a capital C plus is there, then it gives rise to agouti color, which is full color. But this is dominant over C, it is dominant over CC, where you see we like the CH, because this is known as chinchilla, and it gives rise to silver grey color. Then it is still dominant over Himalayan, who is written as CH, and this Himalayan is usually white, and is still dominant over albino, which is pure white, and represented only by small c, small c. That means, see, there are 4 different alleles, but their degree of representation is also varies. That means when capital C are there, it gives rise to agouti color. But when CH are there, chinchilla, it gives rise to silver grey. And when Himalayan nuts are there, white. And when only small c present in recessive form, it is pure white. So this is example. Now let us take example number 3, and this example is from plants. In case of plants, the self-sterility or incompatibility and particularly in case of tobacco, tomato and potato, they are regulated by multiple alleles represented as S1, S2, S3, S4 and so on. Multiple alleles are there. Now if the alleles of the pollen grain and stigma will be same, then there is a rejection. The pollen grain will not germinate. If it will be different, then it will accept and it will germinate. Suppose this is the pollen grain and this is the stigma. Suppose here also it is S1 allele, here also S1 allele. That means it is rejected. That means the pollen grain will not germinate. But suppose here it is S1, it is S2, S3, S4, anyone, then it is accepted. So the incompatibility or self-sterility in case of plants is also an example of multiple allelism. So this is the discussion regarding the multiple allelism. Now let us discuss MCQs. So one MCQ we can take: a certain road accident patient with a unknown blood group requires immediate blood transfusion. His doctor friend donated his blood immediately. What is the blood group of the donor? This is the question, and choices are A, B, AB and O. So what is the blood group of the doctor, you have to tell, and this question was asked in AIPMT. Now the second question. The second question is multiple alleles. Multiple alleles are present, first choice, on different chromosomes, second choice, at different loci of the same chromosome, or third choice, at same locus of the same chromosome, or the fourth choice is on non-sister chromatids. non-sister chromatids. And this question also AIPMT 2015. This is AIPMT 2011. So these are the type of questions which are usually asked from the multiple allelism. I think you have a very clear understanding of the multiple allelism. Thank you very much. God bless you. Have a good day.